Every living system is a chaotic environment. If you measure the exact same biological sample twice, you'll get two slightly different numbers. Cells divide at different rates, protein levels spike, and gene expression naturally wavers in a constant state of flux. This creates a massive problem for researchers. Imagine treating a dish of cancer cells with a brand new experimental drug, and you notice their growth rate slows. You have to ask yourself, did the drug actually work, or is it just random biological variance? When you're staring at thousands of fluctuating data points, your brain is hardwired to find patterns, even when none exist. Human intuition is completely useless here. We need an impartial referee to evaluate the numbers. Extracting genuine biological signal from the surrounding static of natural variance is the first hurdle in data analysis. If you cannot prove your result is real, you cannot claim a scientific discovery. Science operates on a default setting, the null hypothesis. These overlapping distributions represent the assumption your drug did nothing. Any difference is purely chance. The opposing theory, the alternative hypothesis, argues a systematic effect occurred, but it bears the entire burden of proof. The stakes of this framework are intentionally high. You aren't trying to prove your drug works, you're mathematically obligated to disprove the null hypothesis first. By forcing data through this binary choice, researchers ensure any claimed effect is strong enough to beat the odds of random chance. The tool we use to weigh these two hypotheses is called a tTest. It acts as a statistical engine, distilling all the complexity of an experiment into a single calculable ratio. This chart breaks down how that engine works. The first variable is the signal. We calculate this by finding the average or mean of our two datasets, the treated cells and the untreated cells, and measuring the exact distance between the peaks. The second variable is the noise. This is the variance within each group. If your data points are wildly scattered and the curves flatten out, the noise is high. As the data sets get messier, the gap between those averages becomes much harder to trust. The t-test simply takes the signal and divides it by the noise. It measures the difference you found against the inherent messiness of the samples you used to find it. If you have a massive difference between means and very little variance, your t-test yields a high ratio. The math requires a loud signal cutting through quiet noise to grant a valid result. Once the t-test finishes computing that ratio, it generates a specific metric you've likely seen in published papers, the p-value. Look at this distribution representing our null hypothesis. The p-value, shaded here in the extreme right tail, is the precise probability of observing data this extreme if the null hypothesis were completely true. By convention, scientists set a strict boundary called the alpha threshold, typically at 0.05. This represents a maximum 1 in 20 chance that the results are just a statistical fluke. If your calculated p-value shrinks past that 5% boundary line, the probability that you are looking at random variance is low enough that you are allowed to reject the null hypothesis entirely. A p-value of 0.04 does not guarantee that your drug is a miracle cure. It simply provides a standardized mathematical threshold so you can make a confident, actionable decision to keep testing. Because this entire framework relies on probability, errors are a mathematical certainty. Even rigorous science occasionally makes the wrong call. This visual matrix maps the outcomes of our decisions. When you hit a p-value of less than 0.05 purely by chance, you incorrectly reject the null hypothesis. You publish a paper claiming a discovery when you are actually just looking at random noise. This is a type 1 error, or a false positive. Conversely, if your experiment is too noisy, you might fail to hit that 0.05 threshold even when the drug is actually working. You accept the null hypothesis and accidentally abandon a genuine effect. This is a type 2 error, or a false negative. Every statistical test requires researchers to balance these risks. Make the threshold too loose, and the literature floods with false discoveries. Make it too strict, and you might miss a life-saving breakthrough. This delicate balance was manageable when scientists tested one drug at a time, but modern bioinformatics completely breaks the standard model. Today, a single microarray experiment measures the expression levels of 20,000 genes simultaneously. This introduces the multiple comparisons problem. If you test 20,000 separate genes using that standard 5% threshold, the math guarantees you will find 1,000 false positives purely by chance. Your real discoveries will be buried in random noise. To handle massive datasets, researchers use conservative corrections, like the Bonferroni method, which divides that 0.05 threshold by the total number of tests. The requirement for significance becomes severely tighter, wiping out the false positives. Whether you are testing a single cell or sequencing an entire genome, hypothesis testing is an evolving mathematical filter. It enforces a standard of evidence, ensuring that out of millions of fluctuating data points, we only act on the